Hello everyone, welcome to chapter 35 of Campbell Biology, vascular plant structure, growth, and development. So to begin, what's a vascular plant? A vascular plant is a plant that contains a circulatory system, a system that circulates nutrients and water to different areas of the plant. And in this case, most vascular plants have a xylem and phloem, which we'll be talking about and which you may have heard about in your earlier studies. So now, let's begin. The basic morphology, or how the shape and structure of vascular plants, reflects their evolutionary history as land-based organisms that inhabit and draw resources from two very different environments, below and above the ground. You can see here the shoot system and the root system are both, the shoot system is above the ground and the root system is below. They must absorb water and minerals from below and carbon dioxide and light from above. The ability to acquire these resources efficiently is traceable to the evolution of the three main plant organs: the leaf, the stem, and the root, which is this whole system here. And these organs form a root system and a shoot system. So the stem and the leaves form the shoot, and the roots form, the roots. Roots are almost never photosynthetic for this reason. How are you going to photosynthesize if you're not exposed to the sunlight, right? Both systems codepend on each other. The shoot system depends on the water and minerals that roots absorb from the soil, and then the roots need the nutrients and sugar from the plant, from the leaves, in order to not starve. So let's first discuss the roots. A root is an organ that anchors a vascular plant in the soil and absorbs minerals and water and often stores carbohydrates and other reserves. So let's begin. The first primary root, originating in the seed embryo, is the first root to emerge from a germinating seed. So although it's not featured here, the primary root is the first thing that you see break out of the germination. So I don't know if you're familiar with those plant growing videos, but the first thing that emerges apart from the shoot is going to be that main primary root. And that primary root soon branches to form lateral roots that can also branch. So you see here that after their primary root is grown, it starts branching off into these big branches, right? It starts branching off into those big branches, and then those big branches branch off into even smaller branches, and so forth. And it greatly enhances the ability for both. It enhances the ability to anchor the plant and to acquire resources like water and nutrients, as we discussed earlier. Now, what is this long, big shoot-like root in the middle? That's what you call the taproot. The taproot, which usually develops from the primary root, usually only serves, the main purpose of it is for anchoring. The lateral roots usually take most care, the tips of the lateral roots often take most of the role of absorption. So the taproot is mostly there to enhance anchoring. A taproot, although energetically expensive to make, facilitates the anchorage of the plant in the soil. And it prevents toppling, which actually allows the plant to grow taller. So if the plant topples, then it will likely grow to the side like this. So if the plant topples, then it will just start drooping and going down, right? So notably, taproots can also be specialized for food storage, and there's going to be many different types of roots that we're going to see later. So to summarize, roots perform the duties of anchorage and nutrient uptake. And notably, there are two different parts: the taproot and the lateral roots, with the main starter of this being the primary root. Now, let's discuss which one? Let's discuss the stems first. A stem is the plant organ bears leaves and buds. So you can see here, these are the buds, and these are the leaves, right? And the leaves are their own separate organ, which we'll get to later. But before that, each stem consists of an alternating system of nodes and internodes. So a node is the point where leaves emerge from. So you see here the diagram has a node here, but then there's also a node down here and here. And then the internode is the space between them. It's the segment of stem, it's the segment of stem that encompasses them. Now, most of the growth of a young shoot is concentrated in either growing shoot tip or apical bud, which is right here. Apical buds are not the only types of buds found in shoots. You see these little side ones? Those are called the axillary buds, as we see right here. And these can potentially form another lateral branch, a thorn, or a flower. And some plants have stems with alternative functions, like food storage or asexual reproduction. And many of these are labeled as rhizomes, stolons, and tubers. Notably, some of these are often mistaken for roots because they're under the soil, but they are still part of the stem. So as you can see, the rhizome here is a horizontal shoot that grows just below the surface, and vertical shoots emerge from this axillary bud onto the ribosome. So I don't know if you can see it, but you can clearly see the rhizome at least here, this long segment, and these spindly parts are actually the root. And then the stolons, this is a strawberry plant. The stolons are pretty much the same thing as the rhizomes. They're horizontal shoots, but they grow just above the surface. And these stolons actually, they allow the strawberry plant to reproduce asexually, as the plantlets, which we'll get to later with leaves, form a nodes along each runner. So at different points here, here's going to be a plantlet, there's going to be a plantlet, and those will help let them reproduce asexually. And then finally, we have tubers. Tubers, you've all seen the potato, right? Tubers, such as these potatoes, are enlarged ends of rhizomes or stolons, specialized for storing food. So remember that we said that roots can also be modified to, stems can be modified to store food. Tubers are the best example of this. And in fact, the eyes of this potato are the axillary buds that emerge from the stem, and that they mark the nodes as such. So now let's discuss leaves. Leaves consist of, first of all, leaves are the main photosynthetic organ. In addition to intercepting light, leaves exchange gases with the atmosphere, dissipate heat, and defend themselves from herbivores and pathogens. These functions may have conflicting physiological, and anatomical, or morphological requirements. Therefore, in general, a leaf often consists of a flattened blade. So if we go back to here, a leaf typically consists of a flat blade here, so mark this down, and a petiole, or the segment between the point of attachment and then the big blade. And this part, what we're discussing, this part is a node, actually. And remember, nodes often come from axillary buds. And furthermore, when we get to the during this unit, we will see that monocots and eudicots differ in the arrangement of veins in the leaves. So most monocots have parallel major veins, and eudicots generally have a branch network of veins arising from a major vein. So usually for this type of leaf has a branch network of veins arising from a major vein. This is the major vein, the midrib, and thus you can clearly see that it's going to be a eudicot. Now, notably, there's a big difference between simple leaves and compound leaves, which we'll discuss right now. So if we go back here, we see that these leaves are an example of what? These leaves are going to be an example of simple leaves, right? But why are they simple leaves? Let's go back to the diagram. A simple leaf has only one leaf and not multiple leaflets. It has a single undivided blade. So let's write that down. Furthermore, usually some simple leaves are deeply lobed. So you can see the lobes here. Now, compound leaves, in contrast, have many different separate leaflets. These leaflets allow for potentially more sunlight gain, more carbon dioxide gain, but really the difference is just that compound leaves have multiple leaflets at the end of each petiole. And a leaflet has no axillary bud. This is important. So How you can tell compound leaves from simple leaves is that when you see here, there's an axillary bud here. There's no such thing here. Where's our axillary bud? That's how we know each leaf is a leaflet versus like a true leaf. And notably, each leaflet, although it's relatively simple here, it could be divided to even more leaflets. So the main difference is that simple is one blade and compound has multiple leaflets, and that those leaflets are different from true leaves in the fact that they don't have axillary buds. And when we see some evolutionary adaptations of leaves, we can see that leaves have been modified to be thin, like tendrils, so we'll write that down. So the pea plant, if you've seen a pea grow before, it lassos on a support, and it forms a coil and helps the plant support itself upright. And tendrils are typically modified leaves, but some tendrils, like in grapevines, are modified stems. So while here, the pea plant, they're modified leaves, sometimes they might be stems. Other times, the leaves might not conduct photosynthesis at all. These are the spines of the cactus, which a lot of you may have seen before. The spines are actually the leaves of the cactus plant. But however, the stems, the green part, is what actually carries out photosynthesis. Leaves can also be modified for storage. So if you ever peeled an onion, each of those layers is a modified leaf. And finally, they may also engage in reproduction. You see these in the succulent. You can see these plantlets. When we discussed plantlets before, we mentioned that the stolons. Remember, in the stolon strawberry plant, the stolons would allow plantlets to fall off and grow back in the soil and reproduce asexually. So this is an example of reproductive leaves. Now, what are these three vascular plants made out of? I mean, we know that they can be modified. We know that they can adapt. We know that they form the shoot and root system. But what are they made out of? Well, remember in the structural hierarchy, we go from cell to tissue to organ. So since we're discussing plant organs, the next step back must be plant tissues. So we have three basic vascular plant tissues: dermal, vascular, and ground. So let's see. First of all, look at the blue part of this diagram. The blue part is the dermal tissue, which serves as the outer protective coverings, much like your own skin. So let's write that down. And in non-woody plants, so plants that aren't like trees, they don't have bark, in non-woody plants, it is usually a single tissue called the epidermis. And the epidermis in particular is a layer of tightly packed cells, much like, again, our own human skin. In leaves and most stems, the cuticle helps prevent water loss. So in leaves, and in woody plants, protective tissue called periderm replaced the epidermis in older regions of stems and roots. So in a tree's outer layer would probably be tougher. So it would be replacing the epidermis there would probably with periderm. So let's write that down. Now, in shoots, specialized epidermal cells called guard cells are involved in gaseous exchange. And another class of highly specialized epidermal cells found in shoots consists of outgrowths called trichomes. And trichomes are these types of outgrowths, like small hairs that grow out. And Some trichomes may even defend against insects through shapes that hinder movement or glands that secrete sticky fluids or toxic compounds. Again, life has a lot of variations. Now, the chief function of the vascular system tissue is to facilitate the transport of materials to the plant and to provide mechanical support. And the two types of vascular tissues, like we mentioned early in the beginning of the video, xylem and phloem. So, xylem and phloem xylem conducts water and dissolved minerals upward from roots, and phloem transports sugars, which is made in the leaves from photosynthesis, to where they are needed, like in the roots. But before we get to that, we have to discuss the types of plant cells, which we have here. In a plant, as in any multicellular organism, cells undergo cell differentiation. So our brain cells at first are all the same stem cells as before, but as when we're like embryos, all the cells that we have are pretty much the same, right? But after we're born, or even before we're born, actually, they rapidly start differentiating into brain cells, nerve cells, and things like that. Plants have a similar thing too. They have parenchyma cells. They have parenchyma cells, they have collenchyma cells, and finally, it's not listed here, but they have sclerenchyma cells. And then you might be wondering, well, what's the difference between all of them? Let's see. First, let's tackle parenchyma cells. Parenchyma cells have primary walls that are relatively thin and flexible. So you can see here, in comparison to the ones you'll see later, these are very thin. And notably that they don't have a secondary wall either. So when mature parenchyma cells, parenchyma cells generally have a large central vacuole. So remember that a central vacuole is a common organelle in plant cells. And that the central vacuole helps act as a storage tank of sorts. So it stores things like, it could store toxins, it could store food, it could store water. It can just really act as a storage tank for whatever the plant needs. And parenchyma cells perform most of the metabolic functions. This is very important. They perform most metabolic processes. And for example, photosynthesis occurs within the chloroplasts of parenchyma cells in the leaves. Some parenchyma cells have colorless plastids, those in stem and roots, called ameloplasts that store starch. So let's make a little table. Leaves conduct photosynthesis, which I'll abbreviate as PS, via parenchyma cells. And naturally, stem and roots, as they don't really conduct photosynthesis, they most likely have ameloplasts. And those ameloplasts end up as something. Remember we said the central vacuole can store food as well? Well, ameloplasts act as those. They act as starch storages. The fleshy tissue of many fruits is composed mainly of parenchyma. Most parenchyma cells retain the ability to divide and differentiate even under particular conditions. So, in some instances, it is even possible to grow an entire plant from a single parenchyma cell. So let's write that down. Retains differentiability. Now let's discuss collenchyma. First of all, you can see, remember when we said the parenchyma walls are really thin? You can see here that's clearly not the case. Now, what's the difference apart from that? Well, collenchyma helps support young parts of the plant shoot. So remember, the shoot system is the stem and the leaves. Collenchyma cells are generally elongated, so you can't really see this. You can kind of see it when you look at the parenchyma versus the collenchyma. You can see that there's kind of like lines here. And the primary walls are thicker than parenchyma, but they're unevenly so. So you see here, they're not that they're kind of thin, but here they're very thick. They're pretty much a solid gap between them, right? Use a better color. And young stems and petioles often have strands of collenchyma cells just below their epidermis. Remember we said epidermis is the dermal tissue in the three vascular tissues. Collenchyma cells provide flexible support without growth restraint. So let's write that. And finally, at maturity, these cells are living and flexible, unlike sclerenchyma cells, which we discuss next. Now, sclerenchyma, I'll write the name again since it's not here. Sclerenchyma cells also function as supporting elements in the plant, but they're much more rigid, so that's the first thing. And in sclerenchyma cells, the secondary cell wall is thick and contains large amounts of lignin. Now, what is lignin? Lignin is a relatively indigestible strengthening polymer. And basically what it does, it accounts for more than a quarter of the dry mass of wood. It's present in all vascular plants, most likely most of the time in sclerenchyma. And as you can see, since it's in a lot of accounts for over a quarter of wood's dry mass, lignin's rigidity has an important impact on wood. Mature sclerenchyma cells cannot elongate, so that's one thing. They cannot elongate due to their rigid lignin content, but they occur in regions of the plant that have stopped growing in length. Sclerenchyma cells are so specialized to support that many are dead at functional maturity. So let's write that. But they produce secondary walls before the protoplast, so the living part of the cell. New word. That when the protoplast dies, they can still support secondary walls that act as a skeleton that support the plant. And there are two types of sclerenchyma cells. There are sclerids and fibers. And sclerids, which are boxier and thicker, but irregular, often impart the hardness to nutshells and seed coats and the gritty texture of pears. And you can see here in a pear fruit that they're very boxy, irregular, but you can see here in fiber cells that they're much more slender, long, and they're much more organized or uniform in shape. And fibers are usually used for, well, you heard about them. Fibers can be used in clothing, and fibers can be also just used in rope making. That's pretty much it. Now, we finally get back to our reference at the beginning of the video. Xylem and phloem. The two types of water-conducting cells for the xylem, tracheids and vessel elements. So we can see here that these are vessel elements, the ones with these types of divisions in the middle, and these are the tracheids. So what's the difference? Well, first of all, both are dead at functional maturity, so let's write that here. There's so much to write that we have to have an extra slide, right? Become dead at functional maturity. And then I'll make the phloem green. Now, tracheids occur in the xylem of all vascular plants. In addition to tracheids, most angiosperms and a few seedless vascular plants and gymnosperms have vessel elements as well. While the living cellular contents of a trachea disintegrate, the cell's thickened walls remain behind, forming a non-living conduit. So the reason why these look so dried out is because, well, all the parts are living parts, the protoplasts are dead. The cell's thickened walls remain behind. The secondary walls are often interrupted by pits, thinner regions where only primary walls are present. So you can see here, these pits are kind of like almost like gaps. They're places where only the primary wall is present. Now, water can migrate laterally between neighboring cells through pits. That's why they're there. Tracheids, on the other hand, in contrast to vessel elements, are relatively longer and thinner. And water moves from cell to cell mainly through the pits, so you can see here. And vessel elements are generally wider, shorter, and thinner walled. And they are aligned end to end, forming long pipes known as vessels, and in some cases are visible to the naked eye. The end walls of vessel elements have perforation plates that enable water to flow freely. And the secondary walls are hardened with lignin. Remember when we said the sclerenchyma and lignin? This hardening provides support and prevents collapse. Now let's discuss the phloem. The phloem, first of all, is alive at maturity. And before we go into the phloem, let's just write down tracheids. And vessel elements. And so the phloem is, again, alive at maturity. The phloem has sieve tube elements. And unlike the water-conducting cells of the xylem, sieve tube element, in seedless vascular plants, sugars and other organic nutrients are transported through these long, narrow cells of sieve cells. And though alive, they lack pretty much everything about a cell that we know about. They lack no nucleus, ribosomes, no vacuole, or cytoskeletal elements. Now, this reduction in cell content allows nutrients to pass more easily through the cell. The end walls become sieve plates, and they have pores. So we go back to this diagram. The sieve plate here, you can see the many pores that allow the sugar to pass through. Alongside each sieve tube element is a non-conducting cell called a companion cell. Companion cell is right here. And what is this for? This is connected to the sieve tube element, and the nucleus and ribosomes of the companion cell serve not only that cell, but also the adjacent sieve tube element. So now we've solved the mystery of why sieve tube elements can stay alive with such a large reduction in organelle content. In some plants, the companion cells and leaves also help load sugar into the sieve tube elements, which then transport the sugar to other parts of the plant. So now let's look at primary growth and secondary growth. Primary growth, first of all, is growth in length. So always remember that. Primary growth is length, secondary growth is width. And primary growth is made possible by apical meristems at the tips of shoots and roots. So if any of you, if you do AP biology in the future, you may do something with an onion root tip cell. That's the apical meristem. So we can see here that the root apical meristem is composed of, once again, dermal, ground, and vascular tissue. We can see that the bottom has more mature tissues, and the primary meristem continues to remain primary, and that there are also leaf primordia present. Apical meristem cells are undifferentiated. When they divide, some daughter cells remain there and ensure a continuing population of undifferentiated cells. Again, like we said, they have to keep growing since the plant has to keep growing in length, they have to keep some there to remain undifferentiated. And other daughter cells become partially differentiated as primary meristem cells. So we can see here the first cell division, apical meristem, is the very tip, and then it goes to a daughter cell, and then it keeps dividing until it reaches differentiation. But some of them continue to remain back here at the apical meristem. Now, as time goes, as time goes, their growth will go up, and they'll continue. The cycle will continue. The apical meristem will continue to have some cells remain there to undifferentiated, to keep the cycle going, keep growing. As you can see here, a thimble-like root cap protects each root apical meristem. Draw and label a simple outline. So you can see here we have the root cap at the very top with the root apical meristem, the primary meristem, and the mature tissues, which we can see in this section as well. Now, secondary growth is growth in thickness. So let's make sure to label that down. Growth in thickness. It's made possible by two lateral meristems. Now, what are these? These are the vascular cambium and the cork cambium. Now, how each of them grows is very different. But first and foremost, the vascular cambium is more close to the core of the tree, and the cork cambium is more outer. And when a cambium cell divides, sometimes both daughter cells remain the cambium and grow, increasing the cambium circumference. And notably, when a vascular cambium cell divides, sometimes one daughter cell becomes a secondary xylem or a secondary phloem. And usually many more xylem cells are produced. And similarly, when a cork cambium divides, sometimes one daughter cell can become a cork cell. And you can see here that after primary growth is completed, the vascular cambium and the cork cambium start dividing out and increasing the circumference. So the stem thickens as secondary xylem and phloem are added to the plant. Now, the primary growth of a eudicote growth is pretty much the same, but there's a little bit of difference. So, the entire biomass of a primary root is derived from the root apical meristem. The root apical meristem also makes a thimble-like root cap, right here, which protects the delicate apical meristem as the root pushes through the abrasive soil. The root cap secretes a polysaccharide slime that lubricates the soil around the tip of the root, allowing for easier penetration. Growth occurs just behind the tip in 3 overlapping zones of cells, and they're the zones of cell division, elongation, and differentiation. Now, the zone of cell division includes the stem cells of the root, root apical meristem, and their immediate products. New root cells are produced in this region, including cells of the root cap. Typically, only a few millimeters behind the tip of the root is the zone of elongation, right here, where most of the growth occurs as root cells elongate. And even before the root cells finish lengthening, many cells already begin specializing in structure and function. As this occurs, the 3 primary meristems, the protoderm, ground meristem, and procambium, become evident. In the zone of differentiation, cells complete their differentiation and become distinct cell types. The protoderm, the outermost primary meristem, so the blue section, is a single layer of cuticle-free cells covering the root. Root hairs are the most prominent feature of the root epidermis. And sandwiched between the protoderm and the procambium is the ground meristem, and which they give rise to the mature ground tissue. The ground tissue of roots consists mostly of parenchyma cells. And in addition to storing carbohydrates, cells in the cortex, which is right here, and it refers to all this ground meristem, cells in the cortex allow for extracellular diffusion of water. And the innermost layer of the cortex is called the endodermis. So around here, I would say, would be the endodermis. And it's a cylinder 1 cell thick that forms the boundary with the vascular cylinder. Now, the procambium gives rise to the vascular cylinder, which consists of a solid core of xylem and phloem tissues surrounded by a cell layer called the pericycle. In most eudicote roots, the xylem has a star-like appearance in cross-section, and the phloem occupies the indentations between the arms of the xylem star. So when we see here, we see that this is the xylem star that we were talking about, this sort of four-pointed figure here, and that what surrounds it is the phloem. And by increasing the length of roots, primary growth facilitates their penetration and exploration of the soil. If a resource-rich pocket is located in the soil, the branching of roots may be stimulated. And when you see here, lateral roots arise from meristematically active regions of the pericycle, and they actually push through the pericycle. When you see here in this diagram that the lateral root, the formation of it originates in the pericycle and destructively pushes through the outer tissues. And if we look at a real-life example here, we can see that in the buttercup species, we can see that all the parts are there. We can see the star of the xylem. We can see the surrounding phloem as well. Now, the entire biomass of a primary shoot, so if we move on, the entire biomass of a primary shoot derives from its shoot apical meristem. So if we go back all the way up here to the first diagram, the shoot apical meristem is a dome-shaped mass of dividing cells at the shoot tip. So somewhere around here would be the thing that we discussed earlier. So let's go back to the plant's primary growth. You can see that the tips of shoots have something that's pretty much this. You have the leaf primordia, you have young leaf at the sides when we focus on the shoots instead. We have shoot apical meristem, the protoderm, procambium, everything same as we discussed with the zone of elongation, differentiation, and everything like that. Now, the branching of shoots arises from the activation of axillary buds. Because of chemical communication by plant hormones, which is in the future, the closer an axillary bud is to an active apical bud, the more inhibited it is. So let's go back to this diagram. Remember we said that what we call the apical and axillary? Well, this is an apical bud and this is an axillary bud. And the closer in proximity it is to the axillary is to an active apical bud, the more inhibited it is. And that's a phenomenon known as apical dominance. So let's go ahead and define this term. So as the axillary apical distance decreases, goes down, the inhibition decreases. No, sorry, the inhibition increases. And again, this is called apical dominance. And in fact, this is why most people prune their trees and things if they want to grow more buds. Because when you snip off the apical bud, the axillary bud will be free to keep going more, to give rise to more lateral shoots. And if an animal eats the end of a shoot, the chemical communication is disrupted, or if a gardener removes it. As a result, the axillary buds break dormancy and start to grow. Now let's discuss leaf anatomy, or rather before that, stem anatomy. The stem is covered by an epidermis that is usually 1 cell thick and covered with an waxy cuticle that prevents water loss. Some examples, remember, we discussed include guard cells and trichomes. Vascular tissue runs the length of a stem in vascular bundles, which consist of mostly parenchyma, but also may contain sclerenchyma to have some sort of rigid support. Unlike lateral roots, which arise from vascular tissue, lateral shoots develop from axillary bud merostems on the stem surface and do not disrupt other tissues. Near the soil surface, in the transition zone between shoot and root, the bundled vascular arrangement of the stem converges with the solid vascular cylinder of the root. The vascular tissue of stems in most Eudicot species consists of vascular bundles arranged in a ring. The xylem in each vascular bundle is adjacent to the pith, and the phloem in each bundle is adjacent to the cortex. In most monocot stems, the vascular bundles are scattered throughout the ground tissue rather than forming a ring. As you can see here, they don't really form a ring or anything like that. In monocots, they just partition themselves randomly. Now, for leaf growth and anatomy, remember we discussed leaf primordia? Those are projections that shaped like a cow's horns that emerge along the sides of the shoot apical meristem. So if we go back to the figure above, we will see that these are the leaf primordia. Unlike roots and stems, secondary growth in leaves is minor or non-existent. While you don't really grow thicker leaves because that minimizes the surface area to volume ratio. The leaf epidermis is covered by waxy cuticle except where it is interrupted by stomata, which allow for exchange of carbon dioxide and oxygen between the surrounding air. The leaf's ground tissue, called the mesophyll, the middle Greek for middle leaf, consists mainly of parenchyma cells specialized for photosynthesis and has two distinct layers, palisade and spongy, right here and here. Palisade consists of one or more layers of elongated parenchyma cells. Spongy is below the palisade. The vascular tissue of each leaf is continuous with the vascular tissue of the stem. Veins subdivide repeatedly and branch throughout the mesophyll. This network brings xylem and phloem into close contact with the photosynthetic tissue, which obtains water and minerals from the xylem and loaded sugars for transport to other parts of the plant. And each vein is enclosed by a protective bundle sheath, and that's a layer of cells that regulate the movement and substances between the vascular tissue and the mesophyll. So in woody plants, it's a little bit different. Secondary growth consists of the tissues produced by vascular cambium and cork cambium. The vascular cambium adds secondary xylem, wood, and secondary phloem, thereby increasing vascular flow and support for the shoots. And in woody plants, notably, primary and secondary occur at the same time. Now, let's go to tree trunk anatomy. There's not really much to discuss here. It's just describing the layers of rings around the tree. So, in temperate regions, wood that develops early in the spring has secondary xylem cells with large diameters and thin cell walls. And because there's a marked contrast between the large cells of the new early wood and the smaller cells of the late wood, a year's growth appears as a distinct growth ring. And that is how researchers can estimate a tree's age by counting growth rings. So as we can see here, there are many layers to it. In order of inner to outer, they are the bark, layers of periderm, secondary phloem, vascular cambium, sapwood, heartwood, vascular vein, growth ring. And as a tree ages, the older layers of secondary xylem that we discussed earlier no longer transport water and minerals, a solution called xylem sap. These layers are called heartwood. And the newer outer layers are referred to as sapwood because they're able to transport sap. Now, one of the most important parts of this section is really the ABC hypothesis. And the ABC hypothesis specifically refers to the three groups of genes that control the parts of the plant or organ, like they identified the organ that should be there. So there are four whorls in typical flower angiosperm flowers. They're the carpel, petal, stamen, and sepal or sepal. And the three genes that compose them are A, B, and C. If we draw this pretty simple diagram, A, C, B, B, we can see that there are four distinct groups here, right? We have AA, which refers to the sepals. We have AB, which end up signaling for petals. We have BC for stamens. And we have CC, where only carpels are expressed. And as you can see here in this diagram, you can see here that they did it in a different diagram, but the answer is the same. The A controls these two parts, the B has these two, and the C is contained in these two. And when you can see here, the side view of wild-type flowers with organ identity mutations, when one gene is knocked out, everyone is knocked out. That is related to the gene. So, example: if A gene is knocked out, then there will be no more sepals or petals. And I believe that comes to the end of our review of chapter 35. There may be other things that we may not have discussed, but if we do, we will be sure to cover them in a separate